Hello, everyone. We're going to get started. My name is Sarah Berendt, and I'm a PhD student in Rhys Allen's lab in Immunology division. It is my pleasure to be chairing this postgrad seminar. And this is a seminar series which explores the discoveries that have shaped biomedical research and the critical challenges still ahead. I would like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation. I pay my respects to their elders past, present, and emerging, and I embrace their continued connections to this place. We recognize Aboriginal and Torres Strait Islander peoples as the first scientists on this continent who ask questions and especially look to the land, sky, animals, and plants for answers. We respect that as the oldest living generations of teachers, artists, engineers, and scientists, how First Nations peoples understood and continue to understand the world has benefited and will always benefit all of us. The intention behind the seminar series is to give you a chance to gain perspective on the bigger picture of research, hear from experts across our divisions, and engage in conversations that shape the future of science. Today, we will begin by a 30-minute talk by Professors Shalin Naik, followed by a 20-minute panel discussion with experts, Professor Ken Shortman, Professor Kinston Radford, Dr. Cindy Odiger, Dr. Shenbo Zhang, and George Kisoglo. We will finish with a student-only Q&A to wrap up the session. And as a housekeeping reminder for those joining online, please write your questions in the Teams chat or indicate that you want to say something and we'll give you the floor. So today's speaker has been tracking cells before it was cool. From Brisbane to Melbourne to the Netherlands and back, Shalin has followed single cells through the twists and turns of fate, literally. Starting with dendritic cells during his PhD at WEHI with Professor Ken Shortman, then pioneering cellular barcoding in stem cells as part of the Schumacher lab at the Netherlands Cancer Institute. He spent his career uncovering the secret lives of cells. He returned to WEHI in 2014 and was later appointed as laboratory head in the Immunology division, where his lab uses single-cell and clonal systems biology to investigate immunology, cancer, and development. Today, he will talk about dendritic cells and his lab's work in trying to bring them to the clinic. You have the floor, Shalin.
Thanks very much, Sarah. Is this mic working? Yeah. Brilliant. I didn't put the doodad in the doodad. Oh, I'll just do it manually. Thank you very much for coming. And yeah, when Mel Call asked me to help set up one of the first ones of these new format student lecture series, I thought it was a great idea. I love the idea of breakthroughs and bottlenecks because actually that's a reality of what we all face in our career. And it's not just a series of facts, it's actually a series of human endeavors and challenges. So today I'm going to go through some of those breakthroughs and bottlenecks, images care of AI, because yeah, there's not an easy image to describe those two things. So a disclaimer, this is a very WEHI-centric talk for anyone who will listen to this lecture and around the world. You'll think all DC biology was discovered at WEHI. It wasn't, but a lot of it was, especially the early days. I will focus on pre-2010 DC research because as far as I'm concerned, that was really the heyday of the discovery of a lot of the things that we now take for granted. It all happened right here. And really in the lab, in the extended umbrella lab under Ken Shortman, who has retired now, but really it was his work and his team that he built over many, many years that have changed the way we appreciate DCs. And Ken Shortman might not be a name that you know at WEHI walking around when you think about some of the previous big names at WEHI, but you should. Please remember that name because it is worth remembering. Where possible, while embarrassing photos of people are being used. And finally, I do want to talk about new opportunities in the clinic and the focus on our particular approach, but there are many others going on right now. So the immune system, why do we care? Well, this is a painting by Cindy Wright of a hand, a healthy human hand, and we have skin and organs and bones, but actually we're just walking, talking pieces of meat that are 37 degrees and wet, and we're walking around the environment. And if you were to do that ordinarily you know what it would turn into, right? It would turn into rotten meat. So basically, the immune system protects us from becoming rotten meat. If there's nothing else you take away, you will take that away today. And we're on constant alert for viruses, bacteria, parasites, and as it turns out, even cancer. So how does the immune system work? I'm not going to tell you about immunology today, but if you are interested in a basic intro to immunology, I gave a talk in the, pull out your phones if you're interested. Take a picture of this image. I gave a talk in 2016 here in this same format, and I used overhead projectors, and a lot of you may not know what those are, but I used overhead projectors and pens, and I described what all of the cells of the immune system do and how they work together, pitched at about like a university student level. But let's just start with the T cell today. T cell is one of the most important cells of the immune system, and that is a cell that one type can kill infected cells and cancer cells. And so that cell at the bottom there is a T cell, and it's reaching as little fingers into a cancer cell and providing a lethal hit so that cancer cell dies. And these cells are running around your body right now, killing things that shouldn't be there and not touching the things that should be there. And this is the big question. How does a T cell know who to kill and who not to kill? And that was the big question in the 70s. And there were some ideas that maybe it's a macrophage, it does something or whatever. But actually, we found out, no, it is dendritic cells. And what they do is they spy, they have a look for what's going wrong in the environment of your body, and then they educate the T cells. And it was the discovery by Ralph Steinman that actually led to that knowledge. Whoops, I don't know why that disappeared. So if you have a pathogen or an infected cell, or a cancer cell, a dendritic cell can engulf that entity, put it into a really low pH environment, start chopping it up into pieces, into peptides, and they will present a fragment of that thing it encountered on its surface via MHC class 1 or class 2 molecules. Then a T cell comes along and it has a T cell receptor that only recognizes that little peptide. And when you get that recognition, the dendritic cell activates that T cell. It does this through signal 1, which is the antigen itself. Signal 2, which is co-stimulatory molecules, it helps the cell along. And what some people call signal 3, which is the secretion of cytokines that really change that T cell from a dormant, naive, I'm not going to touch anything, to a lethal killer that if it now goes away and sees another cell that has that same peptide, it will introduce a lethal hit. So that's what dendritic cells do. Now, Ralph Steinman discovered them in 1974. He won the 2011 Nobel Prize in Physiology and Medicine. Unfortunately, fortunately for him, he was awarded the Nobel Prize. As you know, it cannot be awarded posthumously, but he died before he found out, just a couple of days. And so he's one of the only people, I think, who were awarded the Nobel Prize who weren't actually there to pick it up because the timing just worked out that he got it, but he never knew he got it, unfortunately. So that was a breakthrough. We found out that DCs are the ones that activate T cells. And there was this assumption that DCs were all the same. But was that the case? And the answer was no. There are actually lots of different types of dendritic cells, including DC1s that are subject of this talk. And it was actually Ken Shortman and David Vremick. These are photos circa 1990. And if you see, is David here today? There he is. All right. Yeah, you can compare hairlines if you want. Those were the great days, weren't they, Dave? Anyway, it was this 1992 paper where they looked by surface facts, and they found this unusual expression of CD8, which was a normal marker of T cells, but actually was expressed on dendritic cells. And so that was a bit weird, and they thought it was an artifact, but they tried really hard. They did all the right controls, and no, it definitely was. And fast forward, and it turns out there's lots of dendritic cells in the body, and these are 4 of them the way I've split them, but actually there's more now, which I won't go into. But basically the ones at the top, the DC1s are responsible for intracellular pathogens in cancer, DC2s for extracellular pathogens and antibody responses, and plasmacytoid dendritic cells for type 1 interferon and antiviral responses. And there's also this oddity of monocyte-derived dendritic cells, which I'll get to a bit later. And they were largely studied in an in vitro setting, although we do know they can exist in vivo. Now, these are some summaries, but they actually can all do a little bit of everything, but a bit specialized. So these are the DC subsets, but what makes DC1s so special? The answer is cross-presentation and cytokines, and I'll talk about what this means. So normally if you're a dendritic cell, or in fact any cell in your body, normally you can present the proteins that are inside your cell through this pathway. You have an intracellular bug, or actually just a normal protein. It's chopped up into pieces by the proteasome, and those peptides are loaded on MHC class 1, and they go to the surface and present that peptide. Now, if a CD8 T cell comes along and recognizes it, and it's a killer T cell, well, that cell will be killed. But in the case of dendritic cells, it has the opposite. It doesn't talk, to kill. It actually talks to get educated. So if this is a dendritic cell and it has an intracellular bug and it finds a naive CD8 T cell, that T cell now gets activated and it can go and kill infected cells or cancerous cells. But if there's an extracellular bug, then it gets phagocytosed. It goes into this phagosome. It gets digested into peptides and is presented via MHC class 2. And that activates a CD4 T cell virus T cell receptor. And that cell goes on to help macrophages and B cells make antibody and those sorts of things. But they are not killers per se. Now, here's the big question. If a dendritic cell is never infected by one of these really important extracellular bugs, or a cell that was infected intracellularly by another bug but never infected the DCs, how could the DC possibly educate a killer T cell? Well, the answer is cross-presentation. And I won't go into it today, but there's a still debated complex process where a phagocytized material can actually enter the MHC class 1 pathway. And DC1s are specialized at cross-presentation. Hardly any other cell in the body does this with the same efficacy as DC1s. And what the result of that is, you see an infected or a cancerous cell by DC1 and it activates a polyclonal T cell response. Yeah, that's a good one. I found this one at Bill. He does not look like this anymore. Anyway, this is Bill Heath and Joe Pooley. Joe Pooley is in the audience. I saw her earlier. Yeah, there she is. And this was one of the key papers. Mike Bevan in the US, but also the labs here discovered that this is how, or this is the unique feature of DC1s. And DC1s used to be called CD8 positive dendritic cells. So if you see that, I use that interchangeably. All right, the other big breakthrough, apart from Jose's hair, was the idea that DC1s take a snapshot. So how is it possible for DC1s, if they encounter a bug at one place, but they have to then travel to the lymph nodes, which might take a day or so to then find the T cell, how can they remember what they saw at the time of infection? And so through a series of neat biochemical tricks, right, cell biology tricks, the DC is able to just freeze what it saw. Testing. All right. Hope people online can hear. Anyway, it takes a snapshot. It maintains what it sees so that even a couple of days later, it's still presenting what it saw at the time of activation. That was a really big development, in my opinion. The other one was that DC1s secrete special cytokines. And it was Hubertus and Meredith that found a lot of these cytokines and the conditions of which they do that. And just for today, I want you to know about IL-12P70 and interferon lambda. These are two of several cytokines that DC1s kind of uniquely produce that we think have really important functional consequences on the immune response. Now, the big question was, where did DCs come from? And that's sort of where I came in 2002 when I came in here. And the work that I did while I was in Ken's lab with Li Wu found that there's a separate developmental pathway. And this is me at the beginning of my PhD. I was clean cut, naive, just eager to learn science. But I had fantastic mentors in Li Wu and Ken Shortman who taught me everything I know. And I'm summarizing the work of my PhD here in this summary slide that we published. And until we had started working on this, everyone thought monocytes were the precursor of all dendritic cells. But that turned out not to be the case, and through a series of experiments, we figured out that monocytes can be the progenitor of a monocyte-derived inflammatory DC. But actually, the pathway of normal DC1s and DC2s was through a different pathway entirely, and PDCs were different yet again. And in fact, it was this monocyte-derived DC that was best reflected by GM-CSF and IL-4 cultures, which many other people around the world had been studying as dendritic cells, inverted commas. But we showed they're actually a very different thing. They're a different beast entirely. And if you want to study these bona fide conventional dendritic cells, you really need the FLT3 ligand axis and can make them in FLT3 ligand cultures. So this represented the first time we could make dendritic cells from a mouse from stem cells in a dish, and I'll come back to that later. But after 4 years of doing my PhD, it had various impacts on my well-being. And that was in part largely to my accomplices at the time. Edwin used to have hair Hawkins, and Nick, look at me, Strum Huntington. Anyway, that was a Christmas party in circa 2006, I think. All right, so this is all in mouse. We were all working on mouse, and honestly, I barely read a human DC paper, which was a real shame because the human DC field was actually burgeoning at the time. But it wasn't until a lot later that we found that indeed humans do have DC1s. We were just looking at the wrong markers. They don't express CD8. For some, accident, we kept looking at CD8, but that was not the right marker. There were other markers that were shared, but CD8 was not one of them. And it was actually Kristen Radford and Derek Hart that really pioneered the discovery and the description of human DCs. And this is from their paper, which in the same issue of JXMed, there were 3 other papers, so a total of 4. And in this News and Views by Jose and Ken Shortman, they kind of go on to describe, it's called Found in Translation, this opinion piece. And it describes the DC1s in human tissue, how to grow them in a dish, right? So they were inspired by the FLT3 ligand, and they found that you could also make human DCs in a dish. And that really changed the game now. Now we could talk the same language and be looking at various things. But from 1992 till now, which was 2010, right? That's a fair bunch of time between discovery of mouse DCs and human DCs. Could it have been taken a shorter time? I don't know. We'll never know. But this is one of those things, I think, in science where unless you explicitly look for it or have the tools to find it, it might be a while before you can connect the dots. So DCs can activate T cells, and T cells can kill cancer. So can DC1s cure cancer? So it's not for one of lack of trying. So people have actually been trying to use DCs for cancer for decades, right? And I'll get to what the attempts have led to, but it's been not much. But for DC1s, the answer is, in mice, yes, they can. But what about in humans? So are DC1s the missing link? So what we do know is that to clear a tumor, and often tumors are heterogeneous, right? They've evolved, they've subclones, they downregulate markers, they overexpress others, and you really need a polyclonal T cell response to kill them. So how can you do that? how can you educate this T cell response? You need an antigen presenting cell. And for a long time, as I mentioned, people used monocyte-derived dendritic cells because they were easy to make in a dish. And they were the first DCs that the kind of Steinman groups used to study from the point of immunotherapy. And there have been hundreds of clinical trials, millions of dollars spent, potentially more, like hundreds of millions. And there's only ever been one FDA-approved therapy called Provenge for the treatment of prostate cancer, and it extended life by only a few months, 4 months or so, and it's not really used anymore. It sent their company bankrupt. So is that the end of dendritic cell immunotherapy? We think the answer is no, and this is just one little graph that represents many that shows that we believe MoDCs are the wrong DCs to be trying to fight cancer, and that DC1s are the right DCs. So DC1s, remember, completely different lineage, completely different cytokines to make them. They are the ones that can take up tumor, they can cross-present, they can present all of the guts of the tumor, all the different neoantigens that are in there to polyclonal T cells to enhance tumor clearance. And this has not come only to our attention, as you can see from these citations. DC1 cancer immunotherapy really is taking off as a concept. So now I'm going to talk to you a little bit about the work we're doing as a group here around, across the team in Australia with the institutes that I mentioned there. So the vision we have is, what if we can give a patient back their own DCs? All right. So a colorectal cancer or another solid cancer patient comes along. They fail their first or second line treatment, and we want to try something new. What if we could take stem cells from their blood? What if we could grow up DC1s in a dish? What if we could arm them in a way that's specific to the cancer using CARs, and I'll get to that in a second. And with this immunotherapy, if we reinfuse it back into a patient, can we actually now get antigen presentation, T cell activation, and tumor clearance? But the big bottleneck has been making enough of them. It's just been really, really hard. With mouse, you can make from bone marrow FLT3 ligand, no problem. But for human, it hasn't been so trivial. And the answer is yes, we can. So we've come up with a new recipe to do that. So Cindy Audiger, who's project lead, Devi and Michael in the lab have figured out a novel approach. Now, unfortunately, due to IP reasons, I can't tell you what that is, but this is what it looks like. So you get more and a lot more, and it's reproducible across multiple GCSF mobilized donors. And now we're talking about from one, we're getting high purity, 10 times high purity, 10 times higher numbers, and kind of between 1 billion and 2 billion DCs. And if you take account of loss through enrichment and purification, we estimate we'll be able to get 800 million DCs about per person for immunotherapy. And now these are the sorts of numbers we think we need to actually have an effect. And until now, to get DC1s, I just talked to someone at a DC meeting last year. She said we needed 6 liters of blood to get 1 million DCs from the blood. They were the numbers they were talking about. So this changes the game in terms of numbers, how we can make them. But tumors aren't pathogens, and tumors can suppress the immune response. So how could we fool the dendritic cells into thinking that a tumor is a pathogen? The answer is dendritic cell specific CARs. So you might have heard about CARs, chimeric antigen receptors. They're used in T cells where this short chain variable fragment can bind a tumor antigen, and it sends a signal to the T cell to kill that tumor. All right, so a T cell sees a tumor, it activates it, it kills the tumor, right? And that's been great in hematological cancer, it's been revolutionary, but unfortunately it hasn't yet had traction in solid cancers. What if we could borrow from that concept? So Shengbo in Steve Nutt's lab came up with this idea, well, what if we engineer the CAR to actually send an activation signal to the DC? Now the DC will activate as though it's a bug, it's a bacteria or a virus or a pathogen, and it'll enhance its engulfment and then its subsequent T cell response. And so the idea is, can we make CAR DC1s? And at least in these preclinical mouse models, the indications are, yes, we can, and they have enhanced tumor clearance properties. So now we've been able to make enough DC1s. We've been able to arm them. What does a clinical trial look like? So with Simon Harrison over at the Centre of Excellence and Jeannie T, who's a colorectal cancer physician, we've come up with this clinical trial that we'd like to start within 5 years. We're calling it the Wildcard clinical trial, weaponized immunotherapy leveraging DC1s or CAR DCs. Anyone who knows me knows I love a good acronym. So I had to look up which words contain DC in the middle. I found Wildcard, then I reverse engineered it, and there it is. So the idea is that a patient comes at the time of diagnosis. They go through screening and eligibility. We perform GCSF, which you inject it and mobilizes the stem cells from their bone marrow. You can manufacture the DCs, and then you give 3 different doses and then see if that works. So we don't know if it will work, but all indications from our mouse preclinical studies is that they will, but one doesn't know until they tries. And I won't go through all of the people on this slide, but it's a huge team that's really working together for a singular goal to try and get DC1s to the clinic. So we'll find out. Hopefully I can come back in here in 5 years and tell you if there's any merit to this approach. I can't tell you much more about this except to say, watch this space. So in summary, DC1s are a specialized DC subset in mice and humans. They have a unique FLT3 ligand-dependent pathway for their development. They cross-present antigen and they take a snapshot of what they encountered. They secrete specialized cytokines to activate the T-cell response, making naive T-cells killers. And they could be harnessed for immunotherapy, for example, with CAR DC1s. And with that, I'd like to thank you all. And I think we might do questions. I'm not sure when. We'll get the panel up now. So I will introduce the panel as you make your way to the stage, everyone, please. So Ken Shortman is still alive and he's here with us today. Ken Shortman. I make that joke only because he makes that joke as well. So yeah, we're really honored to have Ken here. He is retired, but he has been at the Institute. I can't. When did you start here, Ken? I can't remember. A long time ago. Anyway, Kristen Radford from the Martyr Institute, who discovered human DC1s. Cindy Odeger, who's been helping with the translational work to bring DC1s to a GMP setting, and Shengbo Zhang, who kind of designed the cars, and George Kiosoglu, who's a consumer who is involved in this project to kind of help guide from that perspective. Yes, we do need to change. All right. Yes. So Ken, while we're waiting for the screen to change over, so I did hear some when I was at, oops, sorry about that. When I was a young student at some of the conferences, I remember sometimes the work of your lab in characterizing dendritic cell subsets unkindly being referred to as stamp collecting. What would you like to say about that?
Well,
yeah.
Is it on? Yeah. Oh, okay. Well, maybe it was started out that way. It was like accidentally finding different flavors of dendritic cells, and that was like accidental stamp collecting. It wasn't immediately apparent that it might have these implications. So it actually came about because we were studying T-cell development in the thymus. And if you study T-cells, we learned from Ralph Steinemann, you had to think about dendritic cells. And there was this special population in the thymus of dendritic cells bearing CD8. It was the majority population, which is why we found it. And it's only afterwards we looked in the periphery, in spleen, lymph nodes, and found that same population was there. It wasn't just a thymic population, it was out there. And then others in the lab, first of all, Meredith, O'Keefe and Hubertus, found that it had distinct properties. So that's when, for us, it moved a bit beyond stamp collecting to saying, hey, maybe this matters. And then the follow-up finding that it was cross-presenting. So that was it. Certainly at the time, a lot of people said, look, why do you want to mess up a neat concept that's easy to teach to medical students? Conceptually, there's only one cell. Why muck it around? And I suppose the answer to that, my final answer to that, is evolution is damned messy process. It doesn't do things neatly. And so you've got all these strange things, several types of cells doing much the same thing, as it turned out, important subtle differences between them. So that was it. I didn't want to have lots of subsets of dendritic cells either, but they were there. And so it was really seeing that and following it up led us finally to believe, okay, it actually matters. It's not just stamp collecting. There are distinct differences and maybe we can make use of them. Yeah.
Yeah, and when I was here, really the focus was all on mouse dendritic cells. I barely read any literature on human DCs, but at the same time I was doing my PhD, Kristen Radford was working on human DCs. Kristen, can you reflect on what it felt like to be a human DC researcher? How did you enter the field? And then when did you have that aha moment of actually maybe the twain shall meet with human and mouse DC biology?
Yeah, I think for me, it was the end of PhD, and I was making melanoma vaccines as part of my PhD. And it was actually my PhD supervisor at the time, who was Peter Hersey, said, I don't know about you, but I think these dendritic cell things are looking really good. And about that time, that was when the first DC or May DCs were starting to enter the clinic for cancer vaccines. So that's where we sort of came into the piece, and that's where I became interested because they were going to be the latest, greatest immunotherapy. For us, a dendritic cell was a monocyte-derived dendritic cell. And, back in the day, I mean, students weren't willing to give up their spleens or their lymph nodes or their thymus, right? So we couldn't look for these things that you were looking at or studying in mice. So the best we had was blood, really. And so that was the May DCs. And Derek Hart was a really firm believer. He was one of the first to discover the human dendritic cells in the tissues, and a real firm believer that these things that were coming from monocytes weren't the real deal, really. So we started fishing around in blood, but the problem was they're so rare in blood, you had to get a whole leukapheresis product to find the things. So we were talking about, a 20-litre leukapheresis product and a 16-hour isolation day will get you 50,000 to 100,000 cells to play with, and that was a big deal. And that's that seminal publication that we made was made on the grounds of that. So being able to actually start to culture them and study them in more detail has been a game changer, really.
Yeah, that's amazing. Yeah, go on, Ken. You wanted to say something?
One little comment.
Yes.
I do have to thank you, Shalin, and that group of young scientists who decided the nomenclature CD8 positive DC was nonsense because it didn't translate, and to then give them names. And I thank you all for taking my DC and calling it DC number 1.
There was some pushback. In that group, and I stood firm. So you're welcome. Yeah, that's fascinating to see how it all transpired there. George, I wanted to hand over to you now. I'd like you to first tell us a little bit about your background and why you're here, and then what you see as the importance for primary research, and then actually making real efforts to try and take this primary research to the clinic.
Okay, so everyone can hear me. The first thing, I'm here because of WEHI to a degree and Royal Melbourne. The reason for that is I was diagnosed with AML, acute myeloid leukemia, 2013. Had an allogeneic stem cell transplant, 2013. Survival rate 5 to 10%. I'm still here. That's why. While I was going through the treatment, I realised a lot of the reasons I'm here was because of all the research. That's why I came back, or I came to WEHI. And it really highlighted to me the importance of basic science research, translational research, the whole thing. And that's why I like this, but there's a few reasons research are important to me, and a lot of the consumers or patients. Hope is the first thing. I'm going to go back briefly in a history lesson, so you're doing this because, yeah, I'm probably older than most of you here. I remember going back in my primary school days, and I saw a video at the time. I think it was actually a videotape or something, but anyway, and it was about bone marrow transplants, and they measured survival in hours or days. Nothing went further than that. And when you look at that, which was back in the late 60s, early 70s, compared to what you're looking at now, yeah, I know people that have gone through bone marrow transplants that are post 40 years and still going okay. So that's all because of research. Why is this important? When you go through a lot of this, whether it's chemo, whether it's CAR T-cell therapy, anything, colorectal as well, there's a lot of side effects. And you can't always tell from looking at a person about the side effects. One of the big ones people want to get rid of is peripheral neuropathy, because they really don't like living with that. I've been lucky, most of mine have been actually quite mild, but nevertheless, they're still there and they'll probably be there the rest of my life. Not the least of which is the gut and how the allogeneic stem cell and the chemo and everything affects the gut lining, which means I'm on a watching brief over the next rest of my life to make sure I don't get colorectal cancer or anything like that. But apart from that, the reason I think this is really important, when Shalin and when he first introduced it to me going back, I said to him, what are the side effects? And his first answer was, there shouldn't be any.
Shouldn't.
I said shouldn't. And my response was good. I'm still trying to work out how if you change someone's body, there's no side effects, but that's a separate thing. But the point is, if it's minimal side effects and tolerable side effects, it's great. The other thing with this is, if this is a nice, clean, neat way and a less invasive way of being able to help people cure, which is a long way away at the moment, but that's a great thing. And that means we'll have better quality of life, better survival, and everything that goes around that, which is the carers, your family, even the medical profession, because the less you serve us, the more time you've got to see other people.
Yeah, that's brilliant. Thanks, George. Really appreciate your perspective on that. And yeah, George and other consumers have been super helpful in actually helping us realize some of the benefits of what we're doing that, it was staring us in the face, but we didn't understand. And so I think it's really important, if you are, even if you aren't embarking directly on clinical exercise, to actually engage with consumers that are part of your lab. And you can contact the consumer reference group here to try and do that. So now I want to ask Cindy. So Cindy is a postdoc in my lab. Cindy, you were recruited to learn about dendritic cell biology in mice. We never talked about this, yet you're involved. Why and how do you feel about it?
Well, I think it's good. I'm really happy to see that we are moving to a product that may save some people. Moving from the mouse to the human was a bit of a stretch, especially moving from an academic project to a translational project. So when you start academia, when you start your PhD, I guess the keyword, you need to be curious, you need to try, you need, open some door, do an experiment for fun. When you translate, you go for it. And you want to go the shortest, the fastest to the product to be as soon as possible as the clinic. So that was a bit of a completely different way of making the science than what I was used to. But it's fun, and I'm really happy to see that we may be getting there. So I think, yeah, that was good.
Yeah. And what's it like to be working, in academic research? We generally work sometimes in teams, but often just in a silo where in our own brains. What's it been like working as part of a broader team compared to solo?
Yeah, no, that was like, so we are a big team. So I'm looking to work with Michael and David that are there. We have our Peter Mac people with Pat that I see there. So we have been taking advantage of people that have expertise that we don't have. So Christine is she got an amazing mouse model where we can test our human disease. We have been like working in a big team, and that is really nice. So you get exposed to different aspects of what is actually involved to do translational, the GMP production, the safety, the regulation. You need to validate, you need to function, you need to commercialize, you need to think about investors. So it's a big, big new field that englobes different paths. And yeah, so that was pretty good. And everyone, we have a good team. So that's so really why we maybe that's successful.
And what advice would you give to people who are thinking, maybe I'd love to see if this could do something useful beyond research?
Talk to people and go find experts that can give you the answer. And yeah, it's not a one-person project. So go find people that can help you in different aspects in which you don't have the skills. And and come talk to us. Maybe we'll have some cute tips for you.
Yeah. Thanks, Cindy. Now I want to ask Shengbo. So Shengbo, you've also come, to study DC biology, the fundamentals. What possessed you to come up with that idea? And how did you come with that idea? And what has your experience been like going from an idea in your head to actually a concrete demonstration of something?
I think that's very interesting story. Actually, I did my PhD to study the DCs. DC is very hard to culture, to generate. It's very little numbers. Actually, it's one day when I finished the experiment very late. Then I come to a red light. Then I stop in front of that. I think about my experience. And then I ask my question, like, why I stop in front of red light? It's a stupid question, actually. But I do stop, So I think every people stop because you receive two signals. One is you saw it's red, and then you respond to it, right? So you need to recognize and response. But actually, our bodies is also have, different kind of the cancers, bacteria. So we know like the bacteria happen, we recognize it, the DC recognize it, and then it responds to it. But for the tumors, it's different things, right? So first thing, our immune system can't recognize it, right? And then it can't respond to it. So then we thought about an idea, how could we engineer something to make the DC recognize the cancer cells and then give the DC a proper activation signal. Then we come out with the car concept. We talked with Steve and Mikhail about this concept, and luckily they didn't say I'm crazy. But we do generate some proof of concept data and to show this might working. And then we come across with great people and collaborate together. So started to build up a big team, and it's really lucky we can now try to from a simple idea, and then now we can really go to a clinical. Maybe we can do something to the patients. I think that's really great.
That's brilliant. Thanks, Shengbo. I actually didn't know that red light, green light story. Look, I think I'll leave it there and hand over to Mel Call, who's going to not take questions. We can take questions as well. Okay, we can take questions as well. But let's just thank the panel for participating today, especially Ken Shortman, who you just need to understand the gravity of the impact this man has had. It's not been well advertised at WEHI, but I'm going to change that. So please thank Ken and the rest of the panel.
We have some time for questions for the panel, if anybody has any. Good.
Thank you. That's a remarkable work. I'm very interested in your CAR DC1s developing this. So you think what's better about this CAR DC compared to the CAR T or the other therapies?
Shengbo, you can answer that.
I think the advantage of DC is that it's antigen presentation cells. So because it can automatically process the different antigens. So if you think now we all like the artificial intelligence. So if you imagine what is the real intelligence, I think it's DCs. It can tell like what's the heterogeneous antigen and then generates the peptide information present to the T cells. All these kind of the different antigen, DC can recognize and process it. And this is a unique property for DCs compared with the T cells. For the T cells, you need to give the T cells what kind of the antigen you want to target. But for the DCs, we're thinking maybe you just get the DC to take up that antigen and use that DC intelligence. And then you can make the DC to present the different heterogeneous antigen peptides and then activate the immune system to induce a very broad T cell response. So I think that's the advantage of that.
So there's this idea of epitope spreading that what Shengbo just described is called epitope spreading. The idea that you don't have to define what the tumor antigen is upfront like you do with a CAR T cell. The DC is able to learn whatever it is that's different about the cancer compared to healthy tissue and then activate the immune response.
Hey, Cindy. I've got two questions for you, and I think, Shalin, you did show a slide, but what's the timeline like when you isolate stem cells from the patient and give them the CDC? How long does it take, and do they have enough time?
I didn't hear the last part.
Like, is there enough time to give them the DCs?
How long does it take to make them, and is there enough time for the patient?
So that's a really good question. So obviously you don't want 1 month production between you take the cell out and you inject them back. So at the moment we are in a 2-week process. And that is what looks feasible in terms of therapeutic because the patient will go through their first round of treatment. If it's a failure, unfortunately, we have the time to screen them, enroll them, then take the cell and inject back. So that 1 month window is relatively reasonable and is not too long. So we are not putting the patient at risk because in case our treatment do not work, they may end up in another treatment to try to get them. So yeah, we are in a 2-week window from the GCS treatment to the reinfusion for the first dose. Then we think about freezing and reinjecting the second doses as the first product.
Oh, thanks. And second question is there any possibility of off-the-shelf CDC?
Yes. So we are thinking a lot about that. So an off-the-shelf product will allow us to use whatever patient, make the product, put it in a shelf, and wait for when a patient needs it. So not use the own patient's stem cell. And so that will facilitate the product being cheaper and faster. The problem with doing that, we need MHC compatibility. So if we end up injecting MHCs that are not compatible, we may actually do worse than better. So, but we are thinking about it a lot, and we are trying to find how we can explore that option and see maybe by just actually knowing which MHC we can are compatible to make that safe, maybe actually a good way. So yeah, we are thinking a lot about this.
Kristen, did you want to add something there? Or?
Oh, I think in terms of practicalities, you've got cord blood banking, and Australia is very good at public cord blood banking. There's less and less need for cord blood for transplantation purposes, which is why the cord blood banks were originally set up. So there's something like about 12,000 or more units, I think, stored in Australia, but they're also networked worldwide. So that's a huge repository and an ongoing potential for making potential off-the-shelf.
And
therapies.
babies are a renewable resource, aren't they?
Yep.
Hi. That was really interesting to hear. I've just always been curious. So immune cell subtypes are often defined on the basis of their markers, but it's not often clear how that translates to what different things or similar things they're doing in a real person or mouse. So where do you think the line is between sort of different subsets versus just different cells behaving differently based on context or lineage?
Yeah, I might get Ken to answer that, especially because Ken, I was remembering your first forays into RNA-seq of the different DC subtypes and trying to tease apart, well, can that inform function, and what's similar and what's different about the DC subtypes, and how easy is it to demarcate that?
Well, it was very easy to separate them originally in the mouse with CD8 on the surface. Now, I don't know, and I don't think anyone knows what the hell that was there for. No function that we know of. So it wasn't until we had better markers that were more related to function, and one of them was the CLEC9A or Danger 1, whichever you want to call it, that these markers quite clearly separated them. So as far as the surface goes, it wasn't muddy. It was a very clear demarcation. Functionally, the demarcation isn't quite as clear. There is quite a bit of overlap, but certainly the DC1 is the dominant cross-presenter if you don't activate them at the beginning. Other DC can do it if you activate them, but not as well. And so many of the cytokines that are made selectively by DC1 can be made a little bit by other cells, but not as much as well. It's not always black and white. There's just one cytokine that is a clear distinction between them. Others, there's a little bit of overlap. And as I said before, evolution doesn't always give clean, clear answers. It's a bit messy, but there are big differences, big enough to be important.
Hello. Thank you for the great talk and great panel discussions. So I just wanted to ask if this CAR CDC1 can actually guarantee or can help with T cells not getting suppressed in the tumor microenvironment, or if they don't, is there any way that we can address that? Like maybe, I don't know, combining CAR T cells with CAR dendritic cells, or any other ways that you can think of? Yeah. Thank you.
Shengbo, do you want to answer that question?
I think that's a very good question. I think it's quite important if the DC can suppress the T cells or active the T cells. I think that's what we engineered, especially the intracellular signaling domain. We can make it more inflammatory, can make the DC secrete cytokines and then active the T cells. But of course, we can make the DC, because another interesting concept is autologenic DCs. It's very important as well. So we can make that to make the DCs become more autologenic, to suppress the T cell response. So both, I think. That depends on what kind of signaling you design to give it to the cells.
Great, thanks, Shengbo. We have a question online. Justine Minturn has her hand up. Juzzy, do you want to chime in?
Yes. Hi, everyone.
That doesn't work. We'll just go to another audience question.
Hello? Can you hear me?
No? All right, we'll go to an audience question.
Hi. That was an amazing lecture and great panel. I just have a question with the CAR DCs. Obviously, with like cancer treatment right now, there's rarely ever like a cancer treatment that just uses one thing. Usually it has like a lot of other drugs on top of like CAR T cells or like CAR DCs. Like what I'm trying to ask is, how do you see CAR DCs being integrated into like the current treatment that we have for like the cancers, blood cancers, solid cancers, whatever?
Yeah, I think it's quite important because it's quite a new therapeutic approach, but it's not quite new because a lot of people developed like monocyte-derived DC therapy in the clinic, and also the CAR T cells as a cell therapy product. So I think the DC1 cell therapy as a cell therapy product in the later stage, they potentially can, in combination with other therapeutic approach, I think it'd be very beneficial to combined with like chemotherapy to make or radiotherapy to make the tumors release more tumor-associated antigens. That's going to be very helpful. And another major direction is that whether we can use the DC therapy in combination with like anti-PD-1 or anti-PD-L1 treatment. I think all these kind of the current immunotherapy approach could be very useful to gain the function of the DCs.
Thanks, Shagbo. I think we're out of time now. Before I thank everyone, Mel wants the PhD students to stay behind for 2 minutes. Juzzy has a question that CAR DC will be side effect free. You are potentially correct, Juzzy. We do need to examine the possibility that self-antigens are not activated in terms of de novo T-cell activation. Agree. I think natural DCs, the absence of CARs, all data indicates that that's side effect free. But with that, I just want you to all give a round of applause to our panel for today.